Okay, great. So I will proceed then. So first, let's talk about the problem of whole genome homology mapping. So it's a problem of finding all pairs of homologous regions between two genomes. And it can be visualized with a very familiar picture to almost everyone in bioinformatics with a dot plot. So in this picture, we usually have two sequences with coordinate on each axis, and we have a dot on the plot when we have a match. And we usually see long diagonals that indicate a longer homologous regions. So here we see such diagonals, and those diagonals are highlighted on the plot to the right. So our task is to find such diagonals, and as we have longer genomes available, we want to be able to find them in these genomes very quickly and efficiently. So right now, there are several solutions that are able to handle mammalian-sized genomes. First is a very famous Minimap2 that works using a quite traditional approach of chaining of minimizers, where minimizers play the role of those small dots on the dot plot. And the second one is a very non-traditional, well, quite non-traditional approach called MeshMap2, developed by a group of Adam Philippi. And it uses a very different idea. It basically divides the genomes into overlapping windows and then matches those windows with each other using local sensitivity hashing approach. And both of those tools, they can handle mammalian-sized genomes and even larger ones, and they can do this in minutes to hours. However, there are several use cases where running these tools can be quite slow, and one of them is computing the all-against-all comparison of n genomes. In this setting, we have a set of n genomes, and we want to map each genome against each other. And in this case, we have to make a squared order of runs of a pairwise mapper, and each run considers only one pair at a time. And if we have multiple long genomes, this task becomes quite time consuming. It may take multiple hours. And in this case, we have a severe overhead. For example, because each genome is indexed multiple times. For example, if we map genome A against genome B and genome C separately, then obviously we'll have to index genome A multiple times. So we decided to address this problem of homology mapping for a set of genomes using the Bruin graph based approach. So what we do is we build the Bruin graph from all genomes, and then try to find all pairwise chains of so-called bubbles in this graph, and then those chains correspond to homologous intervals that we're looking for. And the advantage is that the compacted de Bruin graph for all genomes can be easily built and stored at once. So let me dive into some details. I will define the de Bruin graph for whole genomes. So for the de Bruin graph, we have a set of sequences. Here we have two sequences, a red one and a blue one, and we have number k. So what we do, we take the first sequence, red one, and write down all subsequences of length k in the order they appear in it. And we designate each subsequence as a vertex and we draw edges from left to right in the order of the sequence of these sequences that we substrings that we call k-mers. And we also repeat this process for the blue one. And as we can see, we have a lot of vertices that have the same labels. And what we can do is we can merge the sequences with the vertices with the same labels together. So we find the vertices with the same label and we merge them. And this way we obtain the Bruin graph for these two sequences for k equals 2. So this graph has some interesting properties. As you can see, the exact matches, they form so-called non-branching paths. Here we have a non-branching graph from TG to GA to AC. That indicates an exact match. And we also have a variation. So we have an inexact match from AC to TC that goes through separate branches. And this inexact match is called bubble. And any homologous region, like haplotype that consists of multiple genetic sites, can be represented in a graph as a sequence of bubbles. And this gives actually the name to the tool bubbles or bubbles. This graph can be compacted to save space. So we can consider all non-branching paths that are highlighted with arcs here and replace them with single edges without losing any information. This is known as the compacted de Bruin graph. And as you can see, it greatly allows us to save space, and it also speeds up the algorithm because we have to operate on a smaller graph. And this object can be constructed very quickly and efficiently, even for long genomes. So we developed previously a tool called Tobacco that can construct this graph efficiently. So this is an example of a graph that's built from 2 genomes. And if we have multiple genomes, we have a more complicated picture, but we still can decompose it into pairwise chains. So let's see an example of a homologous region that is formed by 3 genomes. So here we have 3 genomes, a red one, a blue one, and a green one. And as we can see, if we consider just pairs of genomes at once, we see 3 chains. So if we just consider a pair of genomes, then each time each pair of genome forms this simple object, a sequence of bubbles. And our goal is to, given this compacted de Bruin graph that's formed by potentially multiple genomes, find all pairwise chains of bubbles. So here we see that, for example, the first chain is a bubble that's interleaved by 2 parallel edges, and the second chain and third chain is just a couple of separate bubbles. So we will use the chaining approach. The chaining problem is one of the classic problems in bioinformatics that has been extensively studied since the 90s. And there is an exact dynamic programming solution for this problem, and it has a nice complexity and log-n for pairwise case. However, it relies on a complex RMQ data structure that uses trees and priority queues to answer some queries required in the course of work of the algorithm. So our approach is to limit the gap or the bubble size to parameter b. So in most cases, in most practical applications, we can safely limit the gap size in homologous region to some parameter, and then we can use this limit to use a simpler data structure. So I will not give details about this data structure to save time, but you can read about it in the paper. I will just say that our data structure does not use any pointers or trees or lists. It basically uses a simple bit vector and a special machine instructions that allows us to compare multiple bits at once. And using this very simple and efficient data structure, we can achieve a very high speed up. So I will just give some idea on how chain works. So suppose that we're given strings S and T, a blue string and a red string, and we're given there the Bruin graph G, S, T, K. And what we do, we designate one string as the carrying string, and we consider all k-mers vi of S in sequential order. So we consider k-mers v1, v2, v3, v4, v5 of the blue string S in its order. And during the course of algorithm, we keep a set of chains Q that can be extended by the current k-mer v sub I. So here we have this set Q that keeps all chains that can be extended during the current step. And we all keep only relevant chains in Q. In other words, we keep only chains that within B positions from VI on S, and we update Q as we go over the vertices VI. So as soon as some chains become too far away from the current position, we discard them, and this way we keep the data structure size relatively small. So whenever we consider k-mer v sub I, we have two possible cases. First, we can append the k-mer or vertex v sub I to an existing chain. So here we have a data structure Q that consists of a single chain that forms a bubble in the graph that's shown with dashed lines with the current k-mer vi. And this way, we can extend the chain in Q with this k-mer. And after that, after this step, we add a new chain to the data structure Q. However, it's not always possible, and it could be that there is no chain that can be extended by VI. And this way, we add a singleton k-mer VI to the data structure Q. And this way, we can explore we can find all the pairwise chains of interest in this the Bruin graph. And once we find the pairwise chains, we can project them back to the strings and find the corresponding coordinates of the genomic intervals. So we implemented this algorithm, and we tested on several data sets. So we evaluated along with the competing tools that I mentioned, Minimap2 and MeshMap2, and we used 3D data sets. So we used data set consisting with up to 16 mice genomes. We used a bacterial data set that consists of up to 1600 Salmonella genomes, and we also used some tiny simulated bacteria data set, but for which we know the ground truth. And in all our experiments, we ran our tools with 24 threads. And for Mini and MeshMap2, we basically created pipelines that ran separate pairs of mappings concurrently to get the maximum parallel speed up and utilization of the CPU resources. So I'll show you the results. So here's the running time and memory usage on mice, and we split. We created 4 data sets from the 16 mice data set. We created a data set that consists of 2, 4, 8, and 16 mice. And we ran all these tools on these data sets separately. And as you can see, Bob's is from 3 to 10 times faster than Minimap and MashMap2 in this dataset, but it consumes slightly more memory on large datasets. We also evaluated sensitivity of these tools by calculating recall of protein coding genes. So we downloaded the annotation of protein coding genes on all these 4 data sets. Here I show you recall of base pairs that belong to pairs of orthologous and paralogous genes from the ensemble gene annotation. And for orthologs, like for higher nucleotide, and we pinned all the pairs of genes into pins corresponding to their nucleotide identity. So for higher nucleotide identity, basically all tools have very similar recall values, but for a lower nucleotide identity, BABS has a higher recall. And for paralogous pairs, Minimap2 has slightly higher recall for gene pairs of lower nucleotide identity, and for MashMap2 we did not compute the recall due to technical reasons, because MashMap2 allows filtering of its mappings only in the mode for computing orthologs. So we conclude that Bob's has a sensitivity that is comparable with the competitors. And we also made a similar experiment on salmonella genomes. So we downloaded 1,600 salmonella genomes and created data sets consisting of 200, 400, 800, and 1,600 salmonella genomes, and tried to compute mappings of all genome against every other. And as you can see, in this genome, Bob's is up to times faster, but it consumes a lot more memory because it holds the large index for all the genomes at once. We also created 10 bacterial genome, 10 bacterial data sets that has different genomic divergence. So all bacterial data sets have different distance from genomic distance from the root genome to the leaf ones, and the genomic distance is measured in substitutions per site. So we use this experiment to evaluate how genomic divergence affects recall and precision. And unlike the previous experiments where we used the real data set, here, because we used the simulated data sets, we know the true alignment, so we can measure the recall and precision. So this plot shows that for again, the performance is quite similar for all tools, except that Bob's has slightly higher recall, especially for larger genomic distance, and precision is also similar, except that MashMap2 has slightly lower precision due to the fact that, I guess, it's a probabilistic algorithm and uses windowing schemes that maybe can grab some unrelated, some parts of unrelated sequence at the flanks of the windows. So to conclude, homology mappings can be as chains in the De Bruijn graph using the traditional chaining approach, maybe slightly more efficient data structure. And it allows to achieve considerable speed up to 3 to 10 times on depending on the number and the length of the genomes, and it has comparable accuracy to the competing approaches. And it has potential applications in whole genome alignment and exploratory comparative analysis. So whole genome alignment is especially one of the applications where such mappings of everybody against everybody is needed, and it was basically like one of the main motivations for developing BUBs for us. And would like to acknowledge the funding that allowed this project to happen. And thank you for your attention. We released BUBs. It's freely available at GitHub. And if you want to follow development of these tools, then you can follow me on Twitter. So I'll be glad to take some questions.